Have you ever just stopped to think about how a simple chemical can actually hold all the intricate instructions for a whole human being? I mean, it sounds like straight-up science fiction, but right now, inside almost every single cell of your body, there is a molecule acting as the ultimate digital code. It's a microscopic blueprint that is so complex and so perfectly engineered, it literally governs everything from the color of your eyes to exactly how your immune system fights off a virus.
Welcome to this explainer. Today we are tackling one of the most profound questions in all of science. What is DNA? We're going to break down this incredible molecule by looking at it through a lens you're probably pretty familiar with. We'll explore its physical biophysics, the structure as the hardware of life, and we'll look at its genetic code as the software. By the time we're done here, I promise you'll see your own biology in a completely new life.
Okay, let's dive into this. Here is our roadmap. 1. The molecule of life. 2. The double helix hardware. 3. Rosalind Franklin's photograph 51. 4. The software, base pairs. And 5. Decrypting the genetic code.
All right, kicking things off with Section 1, the molecule of life. Understanding our foundational informational polymer. To connect this seemingly abstract concept right back to your own biology, we have to look at deoxyribonucleic acid, or DNA. This is the primary informational polymer of life. Basically, think of it as the physical medium for genetic storage. Just like a hard drive stores the operating system for your computer, DNA serves as the absolute master blueprint for every single living organism on Earth. It is an incredibly stable molecule that safely houses all the instructions needed to build and maintain you.
And understanding how this molecule works isn't just some academic exercise. It is actually powering modern medicine, especially when it comes to vaccines. For example, in viral vector platforms, like the Oxford AstraZeneca vaccine, we use a harmless virus to deliver a rugged piece of DNA straight into the host cell's nucleus, where it gets transcribed. On the flip side, lipid nanoparticle mRNA platforms like Pfizer and Moderna deliver this delicate, temporary single-stranded mRNA that operates entirely outside the nucleus, out in the cytoplasm. It never even interacts with your actual DNA. So we are literally leveraging the architecture of these genetic molecules to teach our immune systems how to fight off pathogens. It's amazing.
Moving right along to Section 2, the double helix hardware, the physical structure of the blueprint.
Now, what's really interesting about this is how beautifully simple the fundamental building blocks actually are. If you zoom way in, every single piece of DNA is made up of a nucleotide subunit, and that consists of just 3 parts. You've got a 5-carbon deoxyribose sugar, a phosphate group, and a nitrogenous base. The sugar and the phosphate group essentially link together in this alternating pattern to create the sturdy structural backbone of the DNA strand.
But, as you probably know, DNA doesn't just hang out as a single strand, it's a double helix. And for those 2 strands to pair up and lock together properly, they have to run in opposite directions. We call this anti-parallel polarity. Think of it like a flawlessly designed microscopic puzzle. One strand runs from a 5' phosphate group down to a 3' hydroxyl group, and its partner locks right in running the exact opposite way, from 3' to 5'. They are held tightly together by these really strong phosphodiester bonds along that backbone. It's just a brilliant piece of microscopic engineering.
And to really put this into perspective, just look at the mind-blowing scale of these coordinates. In its native state, which is known as bDNA, the physical dimensions are staggeringly precise. The entire helical diameter is just about 2.0 nanometers across. The vertical spacing between each rung on the ladder, only 0.34 nanometers. And it takes exactly 3.4 nanometers for the helix to make one full 360-degree turn. That averages out to about 10 to 10.5 base pairs per complete twist. The sheer mathematical regularity of this structure is honestly breathtaking.
Which brings us to Section 3, Rosalind Franklin's photograph 51, visualizing the unseeable.
So you might be wondering, how did we ever figure out the exact dimensions of something so impossibly small? Well, this number represents a massive turning point in human knowledge. Photograph 51 is the famous X-ray diffraction image captured by Rosalind Franklin and her PhD student Raymond Gosling at King's College London. By beaming X-rays through DNA crystals and basically mapping out where the scattered rays hit photographic film, they revealed this very distinct cross pattern. And in the complex world of X-ray crystallography, that cross pattern was the absolute undeniable signature of a helix.
This whole discovery was essentially a collaborative chain reaction. In the early 1950s, Franklin and Gosling captured that crucial X-ray data. Fast forward to 1953, and using that very evidence, James Watson and Francis Crick successfully proposed the complete right-handed double helix model. And because that physical mapping was finally solved, it paved the way for scientists in 1961 to crack the actual biological code, eventually culminating in a Nobel Prize in 1968.
Let's shift gears to Section 4, the software, base pairs, programming the molecule.
Let's move to the inside and see how this actually builds. Moving past that exterior sugar phosphate backbone we talked about, we arrive at the bases on the inside of the helix. These are the parts that actually program the molecule. Now, these bases are divided into two chemical families. We have the purines, which are bulky double-ring structures. That's your adenine and guanine. Then we have the pyrimidines, which are smaller single-ring structures, cytosine and thymine. In order to maintain that strict perfect 2.0 nanometer diameter all the way down, a bulky double-ring purine absolutely must always pair up with a single-ring pyrimidine.
This creates what we call complementary base pairing, which perfectly matches Erwin Chargaff's famous rules. Adenine always pairs with thymine using 2 hydrogen bonds. Guanine always pairs with cytosine using 3 hydrogen bonds. And here's a cool fact. Because those GC pairs share 3 bonds, they are thermodynamically stronger and much more stable. This means that if you have a region of your genome that's packed with AT pairs, it separates, or literally melts, much more easily under heat or mechanical force.
And this brilliantly illustrates one of the cleverest design features of DNA, the asymmetrical grooves. Because the sugar phosphate backbones aren't attached directly opposite of each other, they naturally create 2 unequal spaces, a narrow minor groove and a wider, deeper major groove. This wide major groove actually leaves the edges of the base pairs exposed. And because of this, regulatory proteins and transcription factors can literally slide right along the molecule and read the software code, the sequence of the bases, without having to rip the hardware apart and unwind the whole helix. How cool is that?
Finally, Section 5, decrypting the genetic code, reading the software.
So, how did scientists figure out exactly how a cell reads this sequence of A's, T's, G's, and C's to actually build a living thing? The monumental breakthrough happened back in 1961. Marshall Nirenberg, Hargobind Khorana, and Robert Hawley conducted this incredibly elegant experiment. They created a synthetic strand of RNA made entirely out of just one base, uracil, over and over again. U, U, U. When they observed the cellular translation process, this pure uracil strand produced a protein chain made solely of the amino acid phenylalanine. They realized they had just read the very first letter of the code. These sequences of bases literally boot up the production of specific amino acids.
So, the crucial point here is that this code is read in specific blocks. We call these blocks codons. A codon is a group of 3 consecutive bases that acts as a highly specific instruction, telling the cellular machinery to add a particular amino acid to a growing protein chain. This microscopic digital code is always read in triplets, just a simple 3-letter code that builds the entire complexity of life.
We started today by asking what the ultimate digital code actually is. And now we know it is a beautifully structured, perfectly precise 2.0 nanometer wide double helix called DNA. By mapping its physical hardware and decrypting its software, we haven't just solved some abstract biological puzzle, we've unlocked the ability to understand our own inheritance, track complex genetic diseases, and engineer targeted life-saving vaccines that protect millions of people. Cracking this microscopic code has undeniably rewritten the future of human medicine. So the only question left for us is, what will we program next?